Hey guys, Miss Cote here, and we are continuing our unit on heredity by tackling genetics. There's a lot to go over, so I've broken this up into 4 separate videos. In this first video, we'll go over the work of Gregor Mendel and how it set the stage for what we know about how inheritance works. So let's get started. A lot of what we know about genetics and heredity comes from the work of Gregor Mendel, that monk who worked with the pea plants. Well, while he was tending to the gardens at the monastery, he made some observations and decided to work with pea plants to investigate further. Why pea plants, you ask? Well, for one, they have short generation times, so you're not waiting very long for the new generation to grow and mature. They also produce a lot of offspring, so you get good numbers. And it was very easy for him to control the pollination of the plants. So there's quite a bit of vocabulary that you'll need to be able to use, so we'll go over them while we discuss Mendel and his experiments. What Mendel was doing was looking at various characters, such as flower color or seed color, any feature that can differ between individuals. And each variant is called a trait. So for flower color, it could be purple or white. For seed color, it could be yellow or green. And unfortunately for Mendel, there were many different varieties of peas for him to study. He focused mainly on 7 characters, shown here in this diagram, which you can pause and look at if you want, but for the sake of time, I'm going to keep going. So for his experiments, Mendel only started with true breeding plants, meaning they only produced the same trait when allowed to self-pollinate, such as one plant only had purple flowers and one plant only produced white flowers. These 2 plants are called the P or parental generation. The offspring produced by crossing these 2 plants are called the F1 generation. F1 stands for first filial. Filial refers to son. And because these plants are a mixture of 2 purebred plants, they're now called hybrids. Mendel then allowed the F1 plants to mate, and the resulting offspring are the F2 generation. So let's see the results of Mendel's experiments. For the first part of the experiment, he crossed a true breeding purple-flowered plant with a true breeding white-flowered plant, and all of the F1 plants had purple flowers. There was not a white flower at all, and when he allowed those purple-flowered F1 plants to mate, the F2 generation produced both purple and white flowers. However, these flowers seemed to follow a pattern where approximately 75% were purple and 25% were white. Mendel repeated not only this experiment, but also did experiments for all of the other 6 characters that he was studying. And no matter what character, he always got approximately the same results. Keep in mind that when Mendel was doing his work, the concept of a gene did not exist yet. So Mendel thought that flower color was controlled by what he called factors. And since the purple flower factor affected the color in the F1 generation, and the fact that you didn't see any white flowers, he called the purple factor a dominant trait, which makes the white factor the recessive trait. All in all, Mendel was a pretty smart guy and was way ahead of his time with his thinking on heredity. With all of his results, Mendel tied it all together by developing a hypothesis to explain that 3-to-1 ratio he kept seeing in his crosses. We're going to use this model to go over his hypothesis, which can be broken down to 4 concepts. First, knowing what we know now, the character of flower color is controlled by a gene. Remember that chromosomes exist in pairs, homologous chromosomes, one from dad, colored blue, and one from mom, colored red. And genes can exist as alternative versions. Mendel referred to them as factors. There was a factor for purple and a factor for white. Those alternative versions are now called alleles. Second, which we just kind of discussed, each organism inherits 2 alleles for each character. One came from dad and one came from mom. The 2 alleles could be identical, but they don't have to be. See how the alleles for flower color in this diagram are not identical. One is the purple allele and one is the white allele. Third, if the 2 alleles are different, one of them will determine what the phenotype will be. In other words, the organism's appearance, in this case, purple. We're going to call this allele dominant. The other allele has no effect on the phenotype. There is no white in this flower. We're going to call this allele recessive. Only one dominant allele is needed to show the purple color. To have a white flower, you would need both alleles to be recessive. And all of these observations by Mendel led him to his first law, the law of segregation, which states that the two alleles for a character are going to separate during gamete formation. So in this diagram, the blue chromosome would end up in one gamete while the red chromosome would end up in the other gamete. You're giving your offspring only one of these chromosomes. They would get the other homologous chromosome from the other parent. Based on what we know now, let's go back to the earlier experiment. We can make predictions about the possible combinations of alleles in their offspring. We do this by using a model called a Punnett square. To represent the alleles, we use letters. Usually the letter represents the dominant phenotype. Because purple is the dominant color, we will use P's in this example. The dominant allele is represented by a capital letter, while the recessive allele is the lowercase letter. So if Mendel started with a true breeding purple flower, the alleles it contains would be two capital P's. When the genetic makeup or genotype has two of the same letters, in this case two capital P's, we call this homozygous, homo meaning same. The genotype has two identical alleles. More specifically, we call this individual homozygous dominant because both alleles are the dominant ones. The true breeding white-flowered plant would contain two lowercase p's, so we would call this parent homozygous recessive. Looking at the gametes these plants would produce, the purple-flowered plant would only be able to give its gametes big P's, since that's all it has, while the white-flowered plant would only be able to give lowercase p's to its gametes. Remember earlier, when these plants were crossed, Mendel found that all of the plants in the F1 generation were purple. Looking further at the genotypes, all of the plants would have the genotype big P, little p. Because there is at least one capital P, all the plants would show the dominant phenotype, which is purple. Genotypically, all plants would contain one capital P and one lowercase p. Because the genotype contains two different alleles, this is called heterozygous, hetero meaning different. Also note that these plants are no longer true breeding. These heterozygous individuals may also be referred to as hybrids, since they are a mixture of the two parents. When these F1 plants produce gametes, they now have two possibilities of what types of P's they can give to their gametes. There's a 50% chance that the gamete will receive a capital P and a 50% chance that the gamete can receive a lowercase p. To see how this gave the F2 generation that 3-to-1 ratio that Mendel observed in his experiments, this is where we use a Punnett square. The possible alleles from one parent's gametes are placed on the top of the square, and the possible alleles from the other parent's gametes are placed along the side of the square. And to be honest, it doesn't matter which parent is where, as long as you have both of them represented. Now to fill in the boxes, you would take the letter from the top of the box and the letter on the side of the box and place both letters inside the box. To make things easier, we usually write the dominant allele first. And there are two different ways you can discuss these results. If you're being asked for a phenotypic ratio, from the 4 potential offspring we got in our cross, 3 of them would be purple while 1 would be white. This is the 3-to-1 ratio that Mendel observed in his experiments. However, if you're being asked for the genotypic ratio, then you would have to look at the letters each individual contains. Out of 4 potential offspring, 1 plant is homozygous dominant, 2 plants are heterozygous, and 1 plant is homozygous recessive. So you can say that they show a 1 to 2 to 1 ratio. One issue that may come up in problems is if a plant produces purple flowers, how do you know what its genotype is? Is it possible that the plant could be homozygous dominant, or is it possible it could be heterozygous? So how are we able to figure this problem out? The answer is that we would need to perform a test cross. Basically, you would take the mystery individual and cross it with a homozygous recessive individual. To summarize this slide, if the mystery plant is homozygous dominant, then all of the offspring would have purple flowers. If some of the offspring have white flowers, then your mystery plant was heterozygous. You may want to remember this concept. It could be helpful when doing problems. Mendel also expanded his work by tracking 2 characters at the same time. Let's go through the example in your book. Here we're tracking both seed color, where yellow is dominant to green, and seed shape, where round is dominant to wrinkled. As always, he started with 2 individuals that were true breeding or homozygous for both traits. One parent is homozygous dominant for yellow and round seeds, while the other parent is homozygous recessive and has green and wrinkled seeds. When these individuals create their gametes, note that 1 letter for each character is put into the gamete. So this parent can only give a big Y and a big R, while this parent can only give a lowercase y and a lowercase r. For this cross, the only possible genotype that can result for the F1 individuals would be this. All the seeds would be yellow and round. Since they are heterozygous for both seed color and shape, they are referred to as dihybrids, hybrid for 2 characters. Next, we would cross 2 individuals from this generation to get our F2 offspring. This dihybrid cross is how we can see if the 2 characters will be passed on together or if they'll be inherited independently from each other. If the 2 characters were inherited together, dependent assortment, then the F1s would create 2 different gametes, capital Y, capital R, and lowercase y, lowercase r, from the original parents. If the 2 characters were inherited independently from each other, then the Y's and R's would not necessarily follow each other. For independent assortment, the F1s would be able to create 4 different combinations of gametes from their genotype. Figuring out possible combinations of gametes is something you need to be able to do. If this is something you're not comfortable figuring out by just looking at the genotype, I'm going to show you what's called the FOIL method. FOIL is an acronym that stands for first, outer, inner, and last. Here's how you would apply this to a genotype combining 2 characters. For first, you would take the first Y and the first R, and that would give you this. For outer, you would take the first and last letter to get this. For inner, you would take the 2 letters in the middle for this, and for last, you would take the last Y and the last R for this. And this is how you would get the 4 possible combinations for the F1 genotype. Going back to the cross, if the 2 characters were inherited together, it's predicted that you would get 2 different phenotypes in the 3-to-1 ratio. However, with independent assortment and 4 possible combinations of gametes, this requires a Punnett square that has 16 squares to it, and the same concepts apply. When you fill in the boxes, you take the gametes from the top and the side. You make sure you keep the Y's together and the R's together, and put the capital letter before the lowercase letter. But when you analyze the phenotypes from this cross, we find that you get 4 possible phenotypes. 9 out of 16 would be yellow and round, both dominant phenotypes. 3 out of 16 would be green and round, 1 recessive and 1 dominant. 3 out of 16 would be yellow and wrinkled, 1 dominant and 1 recessive, just flip-flopped from the last one. And 1 out of 16 would have both recessive traits, green and wrinkled. When Mendel performed these crosses, he consistently got this 9-to-3 to 3-to-1 ratio, which led him to his second law, the law of independent assortment. An extension of the law of segregation, where the alleles will separate during gamete formation, when looking at 2 pairs of alleles, they will also separate independently of each other. The different letters, the Y's and the R's, are not connected to each other. This only applies when the genes are on different chromosomes, or this can happen if the 2 genes are on the same chromosome, but far enough apart where you would see a lot of crossing over happen. If the genes are close to each other on the same chromosome, they're said to be linked and will most likely follow each other and produce different ratios. And this is something we'll discuss in a later video. So as you start to work on genetics problems, the first thing you need to do is to figure out the genotypes of the parents. From there, you can work out what types of gametes they can create. Then set up a Punnett square and fill in the boxes to get the potential offspring. And last, go back to the word problem and see what they're asking you for. Are they asking for genotypes, phenotypes, something else? Obviously, since this is AP Biology, expect some curveballs once in a while. And that's where I'm going to end this video. In the next video, we'll look at other types of inheritance patterns that go beyond the simple dominant recessive inheritance patterns that Mendel was able to explain. And as always, be a good human.